Okay, so in this video, I am going to just basically go through some strategies I used in reading this article, which was the effects of temperature and salinity on the growth of Alexandrium. I'm not going to try and pronounce it, isolates from the Salish Sea. And basically they're just sort of looking at what the influence was of temperature and salinity on how rapidly this particular material or organism grows, because it's something that can ultimately influence toxicity in shellfish, which affects our food supply. So to kind of start out with, I'm just going to kind of go through just the general approach that I took to reading this article. So it starts out, there's an abstract. The abstract kind of provides an overall summary. And then there's the introduction. And the introduction, I kind of read through both of those. And you can see I kind of labeled it. I circled some terms. I sort of identified different key things within the article. Kind of skipped the materials and methods and then went straight to the different figures and started marking them up, trying to figure them out. You'll see that I circle things, I'm labeling the diagrams, I'm writing notes next to it. This diagram, this was printed in black and white, so I color-coded it myself so that I could sort of better interpret the diagram. Again, the next figure I kind of wrote up. I didn't realize that these were correlating to this, so then I identified them as the colors that correlate to the colors. This next one is a table, and it's just data, so I kind of ignored it overall. And then here was another figure that becomes kind of the final figure of their overall article. Once I kind of went through that, then, I kind of come back. I started to read in more detail and started to go through some of the specifics so that then the figures themselves sort of help to tell some of the story. So the abstract itself, when I go through, I kind of identify some things that, sort of contextualizing it in the way that I know what they're talking about, and why we care, right? So again, I'm sort of talking to the text, I'm going through that this is something that's toxic for humans, that they're sort of situating the context of why we would care, so expect that as climate warms, some of these conditions might become more favorable to these kinds of toxic blooms. They use the term hindcast, which is sort of the term I've used in class as retrodict. So again, I'm sort of going back to this, situating the context of terms that I know. If there's anything that I'm not sure about, I underline sort of the key things that I think are worth talking about or focusing on. Over here, I'm sort of noticing that this is sort of part of their conclusions, and this is sort of their, I think that D-1 is per day, and then I confirm that later in the diagram. Over here, they talk about the key index words, so I kind of made sure I knew what those were. HAB, I found in the text, is harmful algal blooms. But basically, this is a little mini version of the paper. So the abstract is a great place to get started with trying to situate and make sure you understand sort of what it is they're going to be talking about. In the introduction now, you kind of get into more of sort of the background information. It provides where it's located, right? So figure 1, I then marked up. So I jumped to figure 1, and I started figuring out, oh, look, there's Lummi Island. That's where we are. So just to try and place the two places that they're collecting data from, the Quartermaster Harbor and the Guaymas Channel, so that I could better understand that. Then I continue in the introduction, and over here, this first sort of section is basically just background knowledge, so some of the citations of other people who've done work. So if I were to follow up and want to do more research, I'd want to kind of look for some of those names. And then it's also building the case for why their research is important. So it's sort of building this, sort of slowly kind of going through and saying, okay, so this is why we care, this is what's happened in the past, that there's been some disagreement in the literature, and so that's how they're settling it for this particular paper. I've underlined some key statements that I think are really valuable to kind of help me, draw me to, if I want to kind of go back and revisit this, these are some of the things that are going to help me remember some of this information. Some of it, if I'm not really sure, there was some sort of, they're talking about the importance of cell numbers, and so I would sort of write it a note to myself to make sure that I could kind of understand what it was that they were talking about there. And then ultimately they're kind of saying, this is sort of the previous work that's been done, and this is how it seems like, so here I'm commenting that, so it seems like there's big ranges, right? So I'm having almost a conversation with myself with some of this text. There's, again, another possible or useful resource along in the context of some helpful information. They also tend to say what their limitations are. So again, the introduction provides and situates the reader with information. The materials and methods I actually went back to as I looked at the figures to help me navigate some of it, but I sort of skipped it at first. We've already looked at figure 1. So then I kind of jumped to figure 2. And figure 2, it really starts to break down, okay, so what are these different units? So again, we're looking at the axes. So mu per day, so d to the power of -1 means it's per day. Mu is the growth rate, so it's mu per day, so it's growth rate per day, and it's a function of temperature. And then each of these in sort of degrees C at these are salinity, and then these are different amounts of temperature settings. So you can see at warmer temperatures, you kind of have these higher growth rates that are happening at sort of these peak locations. They have some equations that help to explain figure 4. We'll come back to figure 3. Because it was black and white, I color-coded it, and that helped me also navigate it. So I kind of labeled where each of these was collected. Basically, I sort of tried to write some notes of where I noted where the highest concentrations were. And again, this is a function of temperature and salinity. So salinity, of course, is in per mil. And then I got to the next figure where then they kind of plot out, okay, so this is the data of temperature relative to our mu, and then this is our growth adjustment relative to salinity. So, and both all these show fit relative to observed. So then they combine all that data into sort of this larger diagram saying, okay, this is where the highest growth rate is relative to temperature and salinity. So this is sort of the final thing that then builds to actually the final one, which is here, which shows over a time frame of months what is sort of your maximum growth, and then where have they been actually categorizing really high levels of algal blooms. And the black were the ones that were actually reported at a federal level, and then the whites were not at the federal level, but they were of dangerousness, but still pretty high. So once I sort of navigated the figures, then I went back to sort of the discussion text. So the methods one, you'll note, is actually a little bit smaller of a font, but then I get to the results where they're talking about it. And I found that now reading through the results, essentially, figure 2 is explained this portion, figure 3 is illustrating this portion, figure 4 is, so really I don't have to read this as deeply because I've already kind of navigated it through the figures. And then in the discussion, it's sort of giving a little bit more context and, essentially repeating some of what we already read in the abstract and the introduction. They're talking about how they were able to replicate the results from others. They're using a model to better understand in order to be able to predict. And then they're comparing their findings to other work that's also come before. Again, they're just sort of building their case. And then ultimately, they're not claiming that it's only one thing. So again, they're using that hesitant language we've talked about in science, but it kind of goes through sort of saying what they think is the most powerful argument for their paper. Again, they're now explaining figure 5, their limitations, and then some final key things that I thought were particularly helpful. And that's essentially the article. And then what I did was, if there was places where I'd want to continue my research, I asterisked those things that I thought might be articles I'd want to follow up on if I were going to continue to do this work. Hope that helps.